Okay, we are in Chapter 3, Three Claims and Four Validities. This is second half, second part of the lecture on the four validities. And as I told you in the first part, the three claims and four validities are the way that Beth Moreling organizes the entire book. So the concepts in this chapter are key for mastering the subsequent materials. So here are the four validities: construct, external, statistical, and internal. These are the kinds of things that if you are designing research, carrying it out, you have to be thinking about. They're problems, and oftentimes they're very difficult to solve. If you are a consumer of research, these are the kinds of things that you want to think about when you're evaluating the quality of research. So next we're going to talk about what each of these things means, and then we'll do some examples, and then we'll talk about when we actually do a study, how we have to trade these things off. It turns out it's very difficult to do all of them at once, and some studies don't even try to do some of them. So without further background, let's go and look at each of them. All right, first one is construct validity. And here we're talking about the quality of measurement. How well do the variables in our study capture or relate to the variables that we're really interested in? So when we measure things, we have some idea that we're trying to get at, but oftentimes it's hard to know if we've got it. And so there are methods that people have developed for examining how well our observed measures correspond to our underlying constructs. So when I'm thinking about this stuff, I like to think about very real, practical, or concrete things that I can see. So we know that different people or different objects weigh different amounts. You know from picking things up, some things are heavier than others. So weight isn't something that you can perceive without interacting with it. You can't just look at something and tell how heavy it is. You think you can, but sometimes you're surprised. So you pick up a box. If it's full of lead, it's going to be a lot heavier than if it's full of feathers, and you might not know which is which. Anyhow, if you want to measure weight, typically you would use something like a bathroom scale or a doctor's scale at the doctor's office, something like that. And what you're interested in are two primary aspects of measurement: reliability and validity. So reliability has to do with repeatability or consistency, and validity has to do with the correlation or correspondence between the observed measures and the underlying construct. So if you think about a bathroom scale, if you stand on the thing and it gives you a reading, you stand off of it and then stand on it again. The second time it gives you a reading, if it's right in the same instance, is going to be pretty similar to what it did the first time. So even if it's a digital scale, it might be off by half a pound or change by half a pound, but it won't change a whole lot. On subsequent days, it might change by a pound or two, and if you wait long enough, it could be 10 pounds or something. But if you just stand on it and stand off and stand on again, the measures that you get are going to be very consistent. If you take a heavy person and a light person and have each of them stand on, stand off, it's going to consistently tell you that the heavy one is heavier than the light one, even if the numbers aren't exactly the same. So that is the sort of measure of a good measuring instrument. It wants to be consistent, and it wants to tell you big numbers belong to big things and little numbers belong to little things. All right, if you compare the bathroom scale to observer judgment, now a person might, let's suppose it's your job to guess how much certain people weigh. If you had 10 people walked by and you had to write down how much you thought each one weighed in pounds, you could do it. How well you did it is sort of up for grabs. So um, suppose that the same people came by again and you couldn't look at the numbers you wrote down the first time. Would you guess the same numbers? Maybe, maybe you wouldn't. If your numbers are different the second time than they were the first, then you have a problem with the reliability. So reliability means consistency or repeatability. Most people, if they're asked to make a judgment about things, will vary a bit from time to time, even if the stimulus or object that they're asked to judge remains the same. And that reduces the reliability of the measurement, of the judgment. Um, the validity, on the other hand, has to do with, are the heavy ones judged heavier and the light ones judged lighter. And observers might be pretty good at that as long as it's pretty obvious. We don't try to fool them, or we don't, cover up some people in, in a tent or something like that. Self-assessment of weight. So we could ask people, how much do you weigh? And if we do that, people may or may not know how much they weigh. If they do, then they may or may not tell you the truth about how much they weigh. So if they tell you the same thing over and over again every time you ask them, well, that will be reliable. If they don't tell you the truth about how much they weigh, then the validity will suffer. So that how much they really weigh will be discrepant from what they tell you. So your observation, your measurement will be off. Okay, so those are the ideas for reliability and validity. Validity of manipulated variables, so the reliability and validity we were talking about for measured variables already. For manipulated variables, what we want to know is, did the manipulation do what it was supposed to? So we want people to feel sad. We show them a sad movie. If it worked, people will feel sad, and compared to, a movie that's either happy or it's just neutral. How would we know if we were successful? Well, we could ask people how they feel after the movie. We could hook them up to some kind of EEG or some kind of physiological measure. We could see whether they cry. I mean, there are various things that we could do to try and see whether the manipulated variable really corresponds to the thing of interest. Okay, but construct validity is about the reliability and validity of the variables that we have in our study. Do they correspond to the thing of interest? If our measure is unreliable, then it won't correspond to the thing of interest. So it has to have reliability. And then in addition, we need it to correspond to the big numbers mean, large things. The small numbers mean small things. So if we're interested in negative affect, then the condition for negative affect should have a lot more negative affect in it than the one that doesn't. Okay. External validity, so second validity. Generalizing from your experiment to the outside. So external validity has to do with how well the results of the study correspond to what you would get if you did it in another context. So if we did a study about drinking behavior in a laboratory, would it actually correspond to what happens in a local bar? Would it correspond to what happens at a sporting event? Many of you have seen the Stanford prison experiment. This was done with college students in a lab at Stanford University. And so the question is, do the conclusions from his study correspond to other groups of people, to other contexts, or is it really kind of universal? So that's what external validity is about. So generalization is just, applying the results to different or broader populations. So we suppose we have students looking at resumes trying to decide whether people are really good applicants. Do you want to interview this person? Do you want to hire this person? Well, we can manipulate characteristics of the resumes and look at how that influences the students' judgments. And we find out A, B, and C. And then the question is, well, if what we really want to know is about hiring managers in companies. The students are not, generally speaking, hiring managers in companies. So how well do the students in the study generalize to hiring managers in companies? So that's what external validity is about. We've got a study, and we have a sample of people and a sample of conditions. So we did some stuff in our study. We want to know, does this really apply to larger groups of people or not? The statistical validity has to do with the numbers and the statistical inference that we make. So I'm going to talk about two kinds of things. One is the statistical errors that come from significance testing, and the second thing we're going to talk about is the magnitude of effect or effect size. So the statistical validity for decision making from statistical significance has to do with making correct and incorrect decisions. So what we do in a study is collect data on a sample, and we use the sample results to make an inference about what's going on in the population. So the simplest way to think about this is a two-group study. So we have an experimental group and a control group, and we want to see if there's a difference between the two groups. So we randomly assign people to the experimental group and to the control group, and then we see what happens. So for example, we might do a randomized clinical trial of a drug. So does this new drug work better than aspirin, or does this new drug help relieve pain better than a placebo, something like that. We get a sample of people and we try our new pain-free, and we see if pain-free is helpful in reducing or eliminating the feelings of pain. Or we might have a therapy that helps people quit smoking. So we run people through two groups. We got a control group in the wait list, and we got the active group who actually does the therapy for no smoking, and then we see after a certain period of time, the number of people that have managed to stop smoking. Or we have a new training program versus the conventional training program for some job task, like how to operate a bulldozer. Wouldn't it be fun? So we've got the new training, we've got the standard training, and we look at how many accidents people have operating the bulldozer, or how much coal they move using the bulldozer over a period of time, or something like that. So we look at the outcomes. All right, so what we're interested in here in the statistical significance is we do the study and we say that the result is significant, which means that there is a difference in the group, so we found something. Or we do the study and we don't see a difference, so we say the result is not significant. Okay, and when we do that, we can be right in two ways and we can be wrong in two ways. And I compare these things to a fire alarm. So, in truth, there can be a fire, and then we've got a fire alarm. So, by analogy, in truth, the drug works or not, but we've just got a local study where we try to determine whether the drug is useful. Or the truth is the bulldozer operator training really is helpful, or it's not in the population, but we just did our little study to try and determine if it is. Okay, so there could be a fire in your house, and generally we presume that the answer is no, there's no fire, but there could be. and either there is or there isn't a fire. Now, you have a fire alarm in your house, and typically they make a lot of noise when they sense smoke. And if there's no fire and the fire alarm is quiet, then it's working properly. If there's no fire and the alarm goes off, that's called a false alarm. And in statistics, that's called a type 1 error. So suppose the training program actually doesn't help people do better job with a bulldozer, but your study says, yes, the training is helpful. Well, that would be a type 1 error. Okay, on the other hand, suppose there's a fire. There's a fire in your house and your fire alarm does not go off. This is really bad. It's called a type 2 error. It's a miss. And this would be like, your training program for the bulldozer operator really is helpful, but you do the study and you can't show that there's any difference between the standard training and your new improved training. That would be a type 2 error. And lastly, suppose that there is a fire and your alarm goes off. That means everything is working. In statistics, this cell is called power, statistical power. So this would be, the truth is your training program really is helpful in operating the bulldozer, and your study shows that, in fact, there is a difference in the groups and the training group does better. Okay, so there's 4 possibilities here, 2 mistakes and 2 correct decisions that you could make. Oftentimes you get a type 1 error if you have one study and you do lots of statistical tests. So by convention, people set the type 1 error rate at 5 in 100 or 5%. And so if you do, 100 of these tests, even if there's nothing going on average, you'll get 5 things saying they're significant. So you'll get 5 false alarms or 5 type 1 errors in 100 tests. Oftentimes the problem with the type 2 error is that we don't have enough people in our study. So it turns out that in order to get statistical significance, you need sufficient numbers of people to make the error of estimation small. And so if you got like 10 people per group, even if there's a pretty big difference, chances are good that you'll miss it. So we need to have enough people in our study that if there really is something going on, that we will see it. And if you get into statistics, then you'll learn about how you do that, how you decide how many people you need. There are well worked out ways of doing that. Right now, it doesn't. You're not in this class. You're not going to have to do that. But what you need to know is that it's possible because of small samples to miss real results. Okay, so that's the first part of statistical validity that has to do with the kinds of mistakes that you can make when you do statistical significance testing. The second part has to do with the strength of association or magnitude of effect. So how big is the effect? If you've got a stop smoking program, it could be really effective so that 90% of the people in the training group or the therapy group quit smoking for the next year. Versus 10% in the control group. That's a really big difference. Or it could be that it's effective, but it's 12% in the therapy group versus 10% in the no therapy group. In that case, you have a pretty small effect. It's there, but it's disappointing. Okay, so it turns out there's a relation between IQ, which is intelligence, cognitive ability, and birth order, but it's too small to be of practical interest. So the studies that have shown this have been like population studies in Sweden or Norway or someplace like that. They get just about all the population and they test them all and they look at the birth order, and sure enough, there's a a reliable, statistically reliable difference, but it's really small. So, if you have a brother or sister that's older or younger, the chances are about 50-50 that they'll score higher on the test than you, and 50-50 that you'll score higher on the test than they do, regardless of who is older. So even though it's there, it's very, very small. It's like the difference between 10% in the no therapy group for smokers and 10.5% or something like that in the therapy group. It's so small that it's nothing to write home about. So on the other hand, when you have a small number of people, you have to have a really big difference in order to be able to show it. So that's the flip side. If you have enough people, a very small difference will be statistically significant. And if you have very few people, you need a huge effect size, a huge magnitude of effect to show statistical significance. So what the bottom line here is, you want to always ask, in addition to saying, is it significant, how big is the effect? What's the magnitude of the effect? And there are a bunch of other things that can go wrong in the statistical analysis. So we've got subgroups, nonlinear relationships, outliers. We'll talk about some of those things later. All right, significance depends on sample size. One of the responses to that is the effect size. And so I already told you what's the magnitude of effect. One way to quantify the magnitude of effect is the correlation coefficient. So we've already talked about that a little bit. And if you had a class in stats, you've seen the correlation coefficient, that little r. And a famous guy named Cohen said, I think 0.1 correlation is small, 0.3 is medium, and 0.5 or bigger is strong. And okay, so you could have a study with a huge number of people and a small correlation like 0.08, and it would be significant, that is statistically significant. And you could go back here and say, okay, well, that's actually a small or weak correlation. So these sort of conventions about how big an effect has to be before you write home. And that 0.08 is significant, but unless we're talking life or death, that's not going to be something you want to spend your money on. Okay, so correlation coefficient is one index of effect size. The second one is called D, or Cohen's D. And Cohen's D is defined as, so the mean in group 1 minus the mean in group 2 over the pooled standard deviation. Correlation coefficient is used when you have a study with continuous variables, both independent and dependent. And the D is used when you have two groups. So you've got the two groups and they're going to have the mean of each group. So we could have the experimental group and the control group. And this SDP, that's the standard deviation within each group, and you just take the average of those, basically. And so this thing is sort of a z-score. So how big is the difference in means compared to the standard deviation of the scores, ignoring the means? Okay. And the numbers are not given here. These are the correlation coefficients. These are, if you take D values, they're about twice as big as these, roughly. So here we got 20 versus 10, 50 versus 30, and 80 versus 50. So these are the values of D, these are the values of R that Cohen says are small, medium, and large. So when you have two means, it's oftentimes difficult to decide, well, how big is that difference? And what Cohen said is, well, why don't we decide how big that is using the yardstick of the standard deviation? And you can see here the same mean difference, but standardized mean difference, D, is larger here than it is here because the yardstick for how big this difference is is the within group variability. So here we've got a D of 0.56 and here we've got a D of 0.24. So in this study, the within group's variability is much larger. So to give you some idea about feeling for standardized mean difference, this is about as big as you would ever see, and this is the vocal tract length of women and men. And so this is the reason that men's voices are deeper on average than women's. And you can see that there are plenty of women with deeper voices than men, but the average difference is pretty big. Difference in height between men and women is about 1.5 standard deviations. Get back there. So 1.4, 1.5 is really big. If you see anything over 1, that's a full standard deviation is enormous. In psychology, typically you'll see standardized mean difference somewhere between 0.1 and 0.3. It's not very big. It might be 0.4, 0.5, maybe if you're lucky. So it's hard to find in most studies. It's hard to find a big effect size. Okay, so we did external, we did statistical, and now internal validity has to do with whether the observed effect is caused only by the experimental treatment. So when I think of internal validity, I think to myself, there can be only one. Only one thing differs across the groups. And this addresses the question of whether your independent variable caused the change and the dependent variable or something else. If there's more than one thing that varied along with the independent variable, then you have confounds. If there's more than one thing that went along with the independent variable, then there's multiple potential explanations of why the dependent variable changed. So psychologists, good ones, can be very, very clever in figuring out ways to make sure that there's exactly one thing that's changing in the study, and that one thing is the independent variable of interest. Turns out to be harder than you would think in order to do that. Okay, so we talked about a bunch of different kinds of things. So we talked about construct, statistical, internal, external validity. All right. So here's your comprehension question. Dr. Scott wants to assess the relations between negative affect and alcohol consumption. He administers a survey to a sample of college students asking about their emotional experience and the amount they drink. In this study, construct validity would refer to, A, how well the negative affect questions on the survey measure negative affectivity, B, how well the correlation between negative affect and drinking summarizes the association in the population, C, how well the study eliminates threat to causal conclusion, and D, how well college students represent people in general. Well, I hope that you can see that each of these 4 alternatives addresses one of the big 4 validities, and only one of those 4 concerns construct validity.